Okay, everyone, welcome back. We're on to the next stage, evolution. Well, essentially, this is a change in the frequency of certain alleles over many generations in order to change the genome of a particular population. There are several aspects of evolution: inheritance, selection, and speciation, and we look at these in turn. Firstly, we have inheritance, and we look at vertical transfer of genetic material. And now this involves passing on genes from a parent to an offspring by sexual or asexual reproduction. In sexual reproduction, of course, the genomes of the parents may differ considerably, and the offspring shows a great deal of genetic variation. Asexual, of course, the offspring are clones of the parents, and the genomes are identical. So in prokaryotes, there's horizontal transfer of genetic material from one organism to another in the same generation. Now, early on in prokaryote evolution, vertical gene transfer was the method for passing on genes to the next generation, and there was lots of gene loss in this process. However, this was compensated for by horizontal gene transfer, which allowed the development of larger genomes and more rapid evolutionary change. However, HGT, horizontal gene transfer, is a bit risky because you don't know what genes are going to be taken up by the prokaryote. Some could be beneficial, however, most are probably harmful. So vertical gene transfer is less risky as an evolutionary method of passing on genes because natural selection plays its part and takes away the deleterious alleles. But as time went on, horizontal gene transfer reduced and vertical gene transfer took over. There was a great deal of variation between offspring, between individuals and offspring and between species, which allowed for distinct lineages to evolve. Now, if we have a look at natural selection, it is, by definition, the non-random increase in the frequency of DNA sequences which increase survival, and the non-random reduction in the frequency of deleterious sequences. This is something you may need to know, particularly this kind of terminology you need to be using at higher level. So if we consider a population of organisms, they show a great deal of genetic variation. For example, let's take these mice, and when they breed, some of them are taken out of the population by having poor camouflage, they're killed by predators. Some starve, that's what a starving mouse looks like. Some die of disease, and some are unable to find lack of shelter. So all these different factors help to maintain the population at a steady level. But only animals with the best characteristics will survive and breed. So if this carries on for many generations, the population, the genomes of the population are going to change. Okay? Those better adapted to the environment will survive and pass on their genes to the next generation. This is essentially natural selection, the driving force behind evolution. So if we just look at a little diagram representing that. So offspring are produced, they show a great deal of genetic variation. They're all struggling to survive within their niche. The better suited ones survive, but the less well suited individuals die. So the ones with the beneficial alleles will survive to breed, and this process will continue generation after generation. Now, this natural selection is often termed as survival as the fittest because the best suited to the environment survive. Let's have a look at an example of evolution. DDT use as an insecticide was quite widespread, and it was very effective initially, but now there are populations of insects that are resistant to DDT. These pests who are resistant to it, of course, have a selective advantage. They're able to survive and breed without competition from those that are still susceptible to the insecticide and are killed off by it. So, for example, there's the black ones are non-resistant insects, and the purple ones are resistant insects. Imagine a population sprayed with DDT. The resistant ones occur quite naturally, not as a response of the use of DDT, but they're just naturally resistant due to genetic variation. So they're sprayed with DDT, and only the resistant ones survive. So they breed to become more populous in the next generation. If there's repeated use of the spray, then that will just wipe out the non-resistant insects, allowing for the resistant ones to be more populous. Okay, so if organisms have deleterious genetic sequences, deleterious alleles, they will be selected against, and in terms of natural selection, there will be a non-random reduction in the frequency of those genes in the population. Any lethal gene sequences will, of course, not survive, and they will not pass on those genes to any offspring. Those with deleterious sequences, of course, will generally produce fewer offspring, and there'll be less chance of passing on those deleterious genes. Now, let's have a look at several types of selection for what's called quantitative traits. Any characteristic of an organism that's controlled by several genes we term as polygenic, and polygenic traits can be measured quantitatively. Okay, so for any particular characteristic, there's usually a normal distribution in the population for different values of that characteristic, as the graph shows here. So that's a typical normal distribution, with the mean and fewer individuals showing more extreme values of that characteristic. Firstly, we look at stabilizing selection. This is where those individuals that show more extreme values for the characteristic are selected against, so increasing the proportion of intermediate organisms within the population. Okay, and this occurs in an unchanging environment. An example, the birth weight of babies. Babies who are born too small generally die of some condition or another. Babies too large, however, cannot be born through the birth canal. So there's a stabilizing selection for babies to be born within the 3 to 4 kilogram range. In directional selection, there's a progressive shift for those individuals representing the mean value for that quantitative trait in one direction or another, away from the original mean. So an example of that would be a black bear size during the Ice Age. They generally got bigger because a smaller surface to area to volume ratio is better at preserving heat. And lastly, we look at disruptive selection, where now extreme variants of the population are favored above the intermediates, and possibly two distinct groups of organisms, each with their own new mean values for that characteristic, begin to form. And that might occur when distinct habitats or resources become available. Examples of that are artificial selection of dogs, for example. We can select for extremely large varieties or extremely small varieties. But disruptive selection could be a driving force behind sympatric speciation, which we'll look at later on. So, species and speciation, of course, what defines a species biologically is organisms that can breed with each other and produce fertile offspring are biologically the same species. Okay? At any given time, a species may be in a stable relationship with the environment. It may be moving towards extinction, or it might be undergoing speciation. So we just like a little graphic to show us that interbreeding individuals within a population. As time goes by, the organisms on the top level, they're becoming new species. In the middle, they're maintaining interbreeding and they're not becoming genetically different. They're in a stable relationship with the environment. Or at the bottom there, those individuals are moving towards extinction. So we know speciation is the formation of new biological species, brought about by evolutionary change. There are two types of speciation that we look at. There's allopatric and sympatric speciation. For allopatric speciation, there's essentially some kind of geographical barrier that splits members of a single starting population. And those geographical features could be things such as rivers, mountain ranges, deserts, or so. In allopatric speciation, first of all, we start off with members of a single population interbreeding freely, so sharing the same gene pool. Now, what happens next is some kind of physical barrier splits the population in two, and those subpopulations can no longer interbreed and exchange genes with each other. Over time, mutations will occur, and this will give rise to genetic variation within each group either side of the barrier. Natural selection will act on those mutations and will favor those individuals with beneficial alleles. Over a long period of time, the two populations become genetically distinct, and if the barrier were to be removed, then you would have two separate species which were no longer able to interbreed and produce fertile offspring. So at that point, speciation would have occurred. Now, in sympatric speciation, it's exactly the same processes basically that occur during allopatric speciation. However, there isn't a geographical barrier that separates the initial population. It's a behavioral or ecological barrier. And of course, we've just seen sympatric speciation may be promoted by disruptive selection. So, in summary, we've learned that vertical gene transfer and horizontal gene transfer have played important roles in evolution. Natural selection, as a definition, the non-random increase in the frequency of beneficial alleles and the non-random reduction of deleterious alleles. Speciation is the formation of new species, and to summarize that, we have an initial population that becomes isolated into two or more subpopulations. Mutations occur over time in those individual populations, and as a result, natural selection will enable those with the most beneficial alleles to survive, and that will lead to speciation. And there's two types of speciation. There's allopatric, which involves geographical barriers, isolating subpopulations, and of course there's sympatric speciation, where the barriers are behavioral or ecological. Okay, thank you very much.